receptor.
Thank you. Good afternoon. I'm really happy to be here today. I'm sorry I have a sore throat, so thanks for the mic. I'm not going to speak about nucleosomes, but I'm going to speak more about the glucocorticoid system that we're using to analyze binding specificity. So first of all, this work is actually the collaboration between an experimental group at the Max Planck Institute for Molecular Genetics in Berlin, Germany. It's the group of Sebastian Meissing. And for the computational part, it's achieved by people from the Vingron department in the same institute where I used to be for my postdoc. And now I move in France to the École Normale Supérieure. And we are sharing some students between the two institutes. So first of all, we're interested in the glucocorticoid. So it's a steroid hormone. And those corticoids are very important. And I'm sure you all know about them, because if you get asthma or some rash, I'm sure your doctor is going to prescribe some kind of cortisone like prednisone. So it's a very important drug. And glucocorticoids are known to have various responses in the body. So they are well known for suppressing the immune response. But they also have other side effects like increasing the circulating calcium. So basically it's removing the calcium from the bones. So it has different effects in different tissue. And it's an interesting model to use in the lab because it's inducible. So the glucocorticoid, the hormone, is going to bind a nuclear receptor, which is called glucocorticoid receptor, so GR. And this receptor is going to bind its transcription factor to activate or repress a target gene. So this is an inducible system in the sense that only if the hormone is present that you can have the binding of the factor to the DNA. So as I mentioned earlier, there are different responses depending on the tissue in the body. And we assessed that by microarray experiments. So we wanted to assess the targets. And we see in blood cells, bone cells, and lung cells that the glucocorticoid actually regulate different target genes. And we actually then wondered if we can correlate that to the different binding of this factor. And we went for ChIP-seq analysis in the 3 different cell lines. And we again see that there is very little overlap in the regions that are bound by this factor. So we also started to analyze in more data those ChIP-seq experiments, and we noticed a real difference between the cell lines. And for example, in the blood cell line, the ChIP-seq peaks are actually heavily biased towards the transcription start sites that are here in the middle, whereas in the 2 other cell lines, there is no specific bias towards this TSS. And we already see that those 3 different cell lines are responding differently to the same factor. So then we were really interested in understanding how the glucocorticoid receptor knows actually where to bind, where to go. So this is the motif of the glucocorticoid receptor. It's an imperfect palindrome, so there are 2 parts with a spacer in the middle. And if you look on the genome at all the positions where we can find this binding motif, we actually see with the ChIP-seq experiments that not all those motifs are bound. For example, here we see that it's bound here, but there are also some peaks for which we actually don't find any GR motif. So we really wondered how the factor knows where to go. And our working hypothesis is that the GR motif itself can be different depending on the conditions, or that it's more like a combinatorial binding where cofactors are important for the binding of the glucocorticoid receptor. So for our experiments, we actually heavily use the RSAT suite of tools. So it's non-line, but it's also possible to install it, a suite of tools that is dedicated to analyzing the regulatory elements. So it has like some tools that are easy, like conversion of formats between matrices or extracting sequences. It's a bit like a Swiss Army knife toolbox, but we actually have also a more complex program like this peak motif program that is used for de novo motif discovery in ChIP-seq data. that is simultaneously running different type of de novo motif algorithms to find overrepresented motif from different criteria. It can be spaced motif, or it can be motif that have positional bias, and it automatically compare the found motif with known collections like JASPAR, and sorry, and then show the results on the genome browser, and also as multiple logo alignment. So we use this program to do motif discovery in our ChIP-seq dataset, and we first focused on the GR motif, which is here, and we actually noticed that depending on the cell line, the percentage of peaks that have the GR motif is highly different. So in the blood cell, we almost don't see the GR motif, only 10% of the peak actually have it, and we see that in the lungs and in the bone, we have a high percentage of peaks that shows the GR motif. So there is clearly a different way of responding to this factor. But we also find a lot of other motifs, whether it's within peaks that contain the GR motif or in peaks that doesn't have them. And those motifs are actually important. So in the lab, Jonas Telorak tested of a peak that is specific to the blood cell line, and he integrated it into a luciferase reporter assay, and he actually showed that this construct is only working if it's inserted into blood cells, and it's not working if it's inserted into bone cells. So biologically speaking, those peaks are actually really cell-specific. And we wondered whether these motifs actually correspond to the binding of GR on motifs that we yet don't know, or on a tethering system, or of a combinatorial approach. And the issue is that the ChIP-seq experiment doesn't give us this information, because the resolution of a ChIP-seq is about 200 to 300 base pair. So, sorry, we wanted to improve the resolution of our data sets, and for this we started to use the ChIP-EXO approach, and the ChIP-EXO approach was developed in the Pew Lab in 2011, and the idea behind the EXO is that it's a bit the same of a ChIP-seq, except that there is an exonuclease that is going to eat, if you want, the DNA from 5' to 3' to actually create a border at the cross-link, at the position where the protein actually cross-links. So you're supposed to get a high resolution view. And this is actually what we see. So here you see in the genome browser our ChIP-seq reads, so in the forward and the reverse strand. And here you can see our ChIP-EXO results. Actually, we didn't really do the EXO ourselves. We sent our samples to the Piconic company that is a spin-off from the Pew Lab. And what you see here is that actually we obtain a much higher resolution, and the signal is much more peaky and restricted. And if we zoom in this region, we can actually see here our GR motif and the reads in the forward strand and the reverse strand that are actually forming a border around it. So we wanted to obtain some footprints, so we start from the peaks. We actually work from our ChIP-seq peaks, because we didn't really want to go into the problem of how to do peak calling on ChIP-EXO. So we started from our ChIP-seq, and we searched for the motif of interest, here GR, and we took a window around it. So for GR, plus minus 30 base pair works quite well. And then we actually look at the ChIP-EXO coverage within those regions, and we do this kind of heat maps. And then if you pile up all the heat maps, you obtain here what we call the footprints. So in blue are the the forward reads, and in red the reverse reads. And this footprint is actually consistent with a dimeric binding of the glucocorticoid receptor. So if you look, for example, here, if it's a dimer, that the exonuclease is going to eat from left to right here and from right to left there, leading two borders, which correspond to this border and this border, but the cross-linking with formaldehyde is actually not 100% efficient. So it happens that even if it's the dimer that is binding only one monomer is cross-linked, and the exo is actually working in terms of cross-linking. So sometimes it will, the exonuclease will eat the DNA from the 5' here, and here, if there is no protein cross-linked, it will go up to this position. So that's why we also see these two other peaks. And the peaks that are the most outwards are actually the highest because they are additive from the monomer and the one that are cross-linked as monomer. So this doesn't mean that it binds as a monomer, but it cross-links as a monomer. And this footprint is actually found even on the actual regions. So you can see here the forward strand that is actually one first big peak and a smaller peak, and here on the reverse strand, one big peak and a smaller peak. So we believe that this is showing the two cross-links and dimeric binding, and this is consistent also to what the Pugh lab had been published on CTCF, which shows the same type of footprints, and they also have two cross-links. We also found other motifs, and I'm going to show you one example out of our analysis. In particular, this motif that we don't know what really it is for at the moment, but we called it the RECOMB motif. And Stefan Starik in the lab actually made a luciferase assay by taking this motif in 3 replicates. And he showed that if you add the glucocorticoid, then you obtain a signal from the GR motif, but also from this motif. So this motif is actually responsive to the glucocorticoid. And this motif can be aligned on the right side here, so it feels like it could be a GR half site followed by this TTCC. And now we looked at the footprint of this motif, and the way we imagine it is, what we did is actually overlaid the two footprints. I hope it's visible. So the plain line are this RECOMB motif, and the dotted line are correspond to the GR motif. And if we look at the right side of what we think is the GR binding, we actually see that here the dotted is the GR footprint, that this RECOMB footprint is actually nicely overlaid. But if we look on the other side of this, what for the moment we call X motif, because we don't know what it is, we see that the footprint is actually does not correspond anymore. So our current efforts are to try to find this protein X that we think is a possible new dimeric partner with the GR. And as I mentioned earlier, our working hypothesis is that the motif of GR would be itself difference, which is our hypothesis for the case before. And we actually recently shown that there are different isoforms for the glucocorticoid receptor, and that 2 different isoforms actually bind on very slightly different motifs. Here, this is the alpha specific, so the alpha isoform, and this is the gamma isoform, and there are just these 2 differences in this position and this position, and this actually affects the way those isoforms are binding. So we believe that the GR motif itself can be important for the regulation by this factor. So by this, I'd like to analyze the people that are working on the glucocorticoid receptor analysis, and in particular Sebastian Meissing, and the people that are working on ERSAT, and in particular Jacques Van Elden, who is supervising the work. And I'd like to thank you.
I have a question.
Yeah.
So now that you have this very high-resolution view of a specific transcription factor, have you compared with the DNase footprinting data, and do you see the footprints in the DNase data that have been previously reported matching?
Yeah, we have started, but we're not yet finished.
Okay.
So I cannot give you the answer exactly right now. Finally, question?
I'm curious if you saw that the additional motif that was novel, that was not recognized, did you look whether it's in any of the databases of selects, PBMs, or
Yeah, so
conserved cross-species motifs?
So when we compare it, we actually see a TID motif, but this doesn't make so much sense with our data set. So for the moment, we are actually this TTCC could also look like a stat family. So we are starting to look more into the stat and trying to, so now we're taking the stat motif, look at the footprint and trying to align these footprints, and we're actually doing it large scale now with the complete JASPAR and other families.
You could also look at the enrichment of that motif in the 300 plus experiments from ENCODE on ChIP-seq.
Yeah,
basically
also.
see if it's a cofactor for any other TFs.
And we actually have it in 3 cell lines, so we also aim at comparing. We also have it in IMR 90, for which there is a lot of data. So yeah,
Great.
it's the beginning. Thank you.